Hello, everyone. Hope you're all doing well. Before I start the lecture today, I'd like to remind you about this table talk about dogs with erect ears and barking, cross with a dog that's droopy ears and silent. This will be on your test, and it involves setting up a dihybrid cross and analyzing the offspring. So make sure that you can successfully fill this out. All right, moving on to today's lecture, we're going to look at examples that are beyond the simple Mendelian genetics, that kind of take this genetics and go into the more complex and really, in reality, what we see more of, where we don't just have two alleles and one is completely dominant over the other, meaning the dominant allele masks the effects of the recessive allele. We're going to look at examples where you might get an intermediate form, okay? So it's not going to be as simple as the genotype and phenotype. If you have the heterozygote of a purple flower and purple is dominant, purple will still be there, okay? Purple will still be the phenotype. So let's take a look. In reality, we have a spectrum of dominance. And alleles will show different degrees of dominance and recessiveness in relation to each other. And the worksheet that you're going to have to do, or the activity after this, makes you really analyze a bunch of different diseases and phenotypes and try to put them on the spectrum. And one thing that you're going to find out that's kind of cool is you also have to look at the organismal level, the biochemical level, and the molecular level, and you might have varying degrees of all of this. Now, what complete dominance is that's what you're used to. One allele, the dominant allele, masks the effects of the recessive allele. So that's on one end of this spectrum. In the middle, we have incomplete dominance, which would be that they share. It's a blending effect. And on the other side, we have a codominance where both alleles get expressed. Now, the best way to learn this is to almost make it where you could give examples of each. So with codominance, let's take the color black, okay? And black would be dominant to white. Well, let's start with complete dominance. The heterozygote for complete dominance would be black, right? Just as you've learned, because the dominant allele expresses, is basically masks the effects of the recessive allele, okay? Now, let's take a look at what an example of incomplete dominance would be. The heterozygote would be in between, okay? So that means that the heterozygote would be a blending effect. So in this case, the heterozygote would be gray. Now for codominance, the heterozygote would be where both alleles are expressed. So if you could have guessed this, codominance would be like, okay, in our example with the black and the white alleles, then the codominant would be maybe a black and white striped. Does that make sense? Okay, so if you want to follow this, one of the easiest ways to try to just get this in your brain is take it through and say, okay, if I have two genes, black and white, and black is dominant over white, if it was complete dominance, it would be black if it was the heterozygote. If it was incomplete dominance, it would be gray. And if it was codominance, both alleles would be expressed in maybe a black and white stripe or a black and white polka dot, something like that. So let's look at these. Again, this is just summarizing, and of course you need to know these definitions. Complete, that's what we've dealt with. Incomplete, the phenotype of the F1 hybrid is somewhere in between the phenotypes of the two parents. Okay, and codominance both have distinguishable, you can see both alleles in some kind of phenotypic expression. Okay, so here's our complete. One allele is completely dominant over the other. These are our famous fruit flies, which you're going to do the virtual lab on. And so this would be where the phenotype of the heterozygote and the dominant homozygote, you cannot tell the difference between them, okay? So that would mean big P, little p, and big P would look the same, all right? Codominance would be the alleles would basically, the phenotype is going to be, so codominance is on this end of the spectrum, in between complete and codominance, we have incomplete. So we're going over here now. Codominance means that each allele gets expressed, okay? In the case of proteins, you would have two different proteins being expressed. An example of codominance, a very easy example to remember, and you do need to kind of try to get these examples in your mind, would be the human blood groups, types A, B, O groups. And I am going to put this YouTube video up that talks about blood groups. That's kind of interesting. Everybody loves to hear about blood groups. And I would suggest that you watch this on your own time. So what you're going to have with blood groups is basically you've got the phenotype of the antigen that gets expressed. So the type A blood will show up with the type A antigen. So it shows up here. The type B blood will show up with the type B on the red blood cells. And the type AB blood is going to have both alleles expressed. So that would be codominance. This isn't a blend of type A and B. Type A and B are expressed, okay? And the two alleles are dominant, and they affect the phenotype in very different ways. And the other blood type that we have is the type O. Now, what happens with these antigens, and you don't really need to know this for your test, you just need to kind of understand that type AB blood is a great example of codominance, meaning that both alleles are expressed. It's not a blending. Both alleles get expressed. What happens when you do blood transfusions is the reason we need to match the blood type is because we need to make sure that your blood doesn't recognize the blood coming in as a foreign body. And so if you have type A blood and you ended up with getting in, a blood transfusion of type B blood, your body would recognize that as something foreign, because it has these antigens on it that it would say that it's not the same type. Your immune system would start to attack it because it would look like a foreign body, okay? If you have type B blood and you put type A in, again, it would recognize the type A as foreign because it doesn't have the little, well, these are antigens, but it doesn't have those little proteins on the outside of it. So it would say, oh, wait, you don't look like me. I'm going to attack you. Now, here's the cool thing. If you're type AB, then guess what? You can only have type AB because if you look here,